Still for question 3, I wanted to show you what else you could have done. What if we didn't get lucky to get already the one gene matching the DNA sequence? Just out of curiosity, let's see what else you could have run. So I put that sequence from question 3 in here, and in my previous video I had here the nucleotide collection, but now I'm going to select RNA sequences instead. So when I selected this, I'm saying I'm only interested in the matches between the sequence that I put there and some RNA sequence from the database. So when I ask this, then I'm saying I only want to see which parts of my query sequence have match an RNA sequence. So that's telling me that that DNA is transcribed, which is what the genes are. So if I select just RNA, let's see what it does and why it's doing that. This one is the result of the one that I did with the whole DNA, not only RNA. And I know that because I look here on the database nucleotide. And this was, in some cases, it's telling me gene. In other cases, it's saying clone whatever on chromosome 1. It's just saying this was sequenced from some clone that they called that. And that chunk came from chromosome 1. But it's not saying what it is. It's not saying it's a gene or not. It just says its sequence came from this. So the same thing. Oh no, actually this one is mRNA. So that one is telling me that it's transcribed. Let's see what else is not very informative. So this one is when I searched the database and I didn't narrow down to mRNA. So let's see if my result for mRNA is ready. Good, yeah. And how do I know that that's the one? I don't have so many windows open. So here, reference sequence RNA. So this one says a bunch of predicted. I'm like, whatever, predicted. I'm not super fond of that. And then we go like, ooh, some interesting ones that are not predicted. Maybe I want to see where the not predicted ones match by sequence. So let's see where, when I have all the 100 sequences selected, my graphic summary is so, and it's like, oh, it looks like there are multiple genes. Looks like, so there is this gene here, another one here, another one here. Yeah, well, 3 genes. So this one would be, okay, that predicted, this one predicted. Okay, so then I might go like, all right, I want the ones that we have biological information for. So I'm going to unselect this and I will select only the stuff that doesn't say predicted. And then you can see here, see how there's variant 1, 2, and 4. This might come from alternative splicing. Let's see if they match the same spot on the genome. 1, 2, 3, 4. Good, all four variants. Okay, this is from something else. It's not humans, but it does say mRNA. It doesn't say predicted, so I'm interested in that. Okay. So let's see where those genes are predicted. Oh, look at that. So what does this thing mean? So this is my query sequence. So that's the sequence I entered. And then these are the mRNAs that match that. And so this thin line here says that this part of this hit doesn't match this part of the query. And it could be because this sequence is different from that, or it could be that this sequence doesn't exist in the mRNA. It only exists in the DNA. So this could be well, in here it's nothing, but in here it would be an intron. So this part here of the match, show me the little hand, there you go. Okay, that's that gene that we even know the function for. And it's going to say the same thing on this one. Come on, just show me. Okay, there you go. So is that gene? So I knew it was the same because it has this line connecting, but this is a different hit from this. But these are the different variants of that, the human gene. So there will be alternative splicing, meaning in one version, this whole piece is going to make to the protein. In another version, only this part will. So this little difference here was an exon in this variant and not in this variant. So let's see, this is, yeah. So there are all the different variants for Homo sapiens. And I'm betting that the last one here is the other one, the different species. Yeah, there you go. Oh, aren't you glad when stuff works? So there is only one gene predicted, and it's the same gene as we found when we did the BLAST N. And the parts that match to the mRNA are not the full sequence, but the answer is still, there is one gene that is predicted in that sequence that I entered as a query.